What's up, everyone? Welcome. We are live right now on Cannabis, and if you're joining us on Spotify or your podcast listening feed of choice, thank you so much for joining us. We appreciate it. We have a great episode for you today. This is another episode for growers. We're going to be talking all about mycorrhizae with Colin Bell, Dr. Colin Bell of Mammoth Microbes fame. You may have heard him. We've been chatting with him for years and years and years. He's back again, going to be chatting with us about mycorrhizae. So JR has a bunch of questions ready for us today. But before we get into it too far, we need to make some shout outs. First off, shout out to everyone in the Cannabis community. You can search Cannabis in the App Store on iOS, or you can go to cannabis.app in your browser, search Cannabis, and use the code BUZZ. And that will give you your first month free. You can join us for as little as $4.20 a month. Your support is very much appreciated. And once you join, there's a sign-up form and the pin posted up at the top. Fill that out, and you may get a pack of seeds in the mail someday. So join us. We'd love to see you over there. And then a couple more shout-outs real quick. LostCoastPlantTherapy.com. We really appreciate them. It's an organic IPM spray that you can use to spray your plants and help take down some bugs, take down mold. I've just sprayed it actually this morning because we're dealing with a bunch of moisture issues right now on the West Coast, and I'm trying to fight back botrytis and things like that. So check out LostCoastPlantTherapy.com. We appreciate their support. And then last one is sacred3mushrooms.com or tanazigardens.com. Use the code Cannabis on there. You can get your own fungi home grow kit. He's got everything you need, the grow container, the medium, everything you need, which can be hard to track down in some cases. So check that out over at his website. Use the code Cannabis for a discount. And then I'm going to throw it over to JR.
Thanks, Q. Today I'm going to do some selfish plugs today. I've been developing this Dit's Amazing Botanical Rub, and I'm finding a lot of people really getting a tremendous amount of relief and help. It promotes pain control, anti-inflammatory, and it also promotes vascular healing. So it gets blood flow to areas that are injured. It's great for post-workouts. It's a fantastic way to treat your body. I have it available on my website, Tokens Roots. I can't really say the rest of the address because they, get mad at me about stuff like that. But you can contact me and get more information if you need it. And I really want to jump into this episode and kind of hammer this one out because we've got a lot of really good information to cover. And we have someone who spent years and years in the field studying this, not just from a bacterial perspective from products in the past, but his actual field work in the field itself. And we're going to be talking about endo and ecto mycorrhizae, and we're going to be able to talk to someone who's really, really studied it in a professional educational setting. So we're happy to have Dr. Colin Bell onto the show to kind of break it all down for us. And we're going to hammer through this one pretty quick, folks, to try to cover a lot of information. So welcome to the show, Colin.
JR, Q, thanks so much for having me back on the show. I appreciate it. I saw the questions that you sent over, and I was just thrilled to be able to read and kind of talk fungi. You talked about it years ago when I was a graduate student working at Texas Tech. And for years and years, we just recovered soil samples and soil organic matter samples and cultivated fungi from them and studied that through all my research training. Obviously, these are very relevant in the professional setting and in agriculture. And so it's really fun to be on the show and talk fungi.
Sweet. Well, let's get into it, brother.
Yeah.
So, is there genetic evidence linking current fungal lineages that we have scattered all over the planet to a ancient supercontinental system where we kind of had this Pangea thing going?
Well, there's evidence. Scientific evidence is subjective to the data you collect over time. there is plenty of DNA evidence that suggests there were fungi that existed way back. We're talking hundreds of millions of years ago, quite honestly. How we think about fungi is Earth started in some kind of aquatic world. Fungi, for the most part, are not aquatic. There's one, amphibian pathogen, oomycetes, that's aquatic, but it has nothing to do with the fungi that we think about in soils. It's a very different morphology and type of fungi. But fungi were the first microorganisms that started to colonize the land and create a habitat, and really closely with plants to terraform the earth, to allow for higher life to grow. So we find plenty of evidence of DNA. One of the things that separates these super lineages that extend, we see groups and broad groups of fungi, basidiomycetes, ascomycetes, which are very large kingdoms of these fungi. What I'd say though is because of the quick adaptation of microorganisms, I wouldn't say that they have found the exact same strains or species, but they see ancestral. So it would be kind of like the dinosaurs of the past, and we see traces of them in current life. The same type of concept would exist with the fungi.
And would that include Antarctica?
I actually looked this up because I was trying to give you a good answer here. I think that, whatever they study, where the data is, which you would find, but the Antarctica and Earth in general was very different. And, you think about the supercontinents and as they shifted, and life has shifted, and the continents have shifted significantly. And so to say that Antarctica was in the place it was when life first existed on Earth would not be a true statement. And so there's very different ecotypes and environments. But so, kind of life covered one system, and then the continent started to divide. And so you'll find traces of that. But I didn't find a lot to speak too much onto the evidence I find. But what you're going to do is you're going to mine it, and you're going to find DNA life, especially the early ancestral bacterial single cell organisms, and then the fungal, the eukaryotic traces, which is the next higher life form in kind of the tree of life, as life started from a bacterial world, a chemical world, to a bacterial world, to a eukaryotic fungal world, quickly to a plant world, and then beyond. You're going to find those traces of microorganisms on all the continents.
So with our show being about cannabis plants, they typically we talk about how they have like a relatively shallow root system, right? So
Sure.
is the cannabis plant, when it's tapping into the mycorrhiza or the hyphae network, is it tapping in, is its roots kind of getting to tap into things that are beyond its reach, so to speak?
Yes, and I think that to the extent that occurs depends on the environment the cannabis plant's grown in. Absolutely, positively, 100%. If you're in an outdoor growing environment, that's what these fungi do. We can talk about, we're going to talk about free living fungi. We're going to talk about symbiotic fungi. Free living fungi, typically called the saprobes, and we can talk about why they're free living, and we could talk about more of the symbiotic groups of fungi, which are the mycorrhizal fungi. And the mycorrhizal fungi consist of arbuscular mycorrhizal fungi and then ectomycorrhizal fungi. So those are the three groups of fungi for the most part we'll talk about today. But any of the symbiotic fungi, they're going to associate with roots somehow, ectomycorrhizal or the arbuscular mycorrhizal, and they do extend past the roots. And so they actually quite literally extend the capacity of the surface area to exposing, they create more surface area within the soil or substrate for the plants to actually mine and take advantage of. So we know that plants absorb from the soil. If there's more out there, more surface area that it can explore, there's more than it can absorb.
And so now it's running into glacial rock. Is it able to actually mine into that rock and start extracting?
Yeah, well, it's probably not running into glacial rock everywhere, but that's a great example of how capable fungi are of penetrating or creating habitats out of ecosystems. Let's just think about rock. Let's think about concrete. Let's think about a tree root or anything like this. Fungi do a really good job of penetrating and existing and breaking apart and delving into the root zones, even from compact soil perspectives. They do that a couple of different ways, but I think the way that fungi are most capable of doing this, and maybe we should talk about the architecture of the hyphae. Hyphae are
Yeah.
basically kind of like roots. You can think of them like roots, except they're a little more microscopic. You can see them, but they're just tubes. They're like hairs. Honestly, you can think about them like horse hairs. And a single strand is called hyphae, and a group or a cluster, maybe that you might see when you see cheese mold or bread mold, and it's kind of in a group or a circle, that's called mycelium, where it's a cluster of hyphae that is conglomerated together. Either way, those are transport tubes, and they extend apically and grow just like plants do from the tip, and they continue to extend out. And they're really good at transporting nutrition and hydration, water, through that tubular structure. So it's like a network that it can transport ions, it can transport types of water, and it can penetrate in the most. And I wanted to explain that because the way it penetrates rock, even glacier rock over time, two main ways is it creates turgor pressure, just like roots can almost bust up a sidewalk. And you've seen that happen, can grow into rock. Well, it creates turgor pressure just like plants do to allow plants to stand up above ground, and it allows plants to penetrate their roots deeper into the root surface. Fungi act the same way. And so they can create turgor pressure or hydropressure and push and start to dive in between the cracks and start to break that apart. The other mechanism coupled with that is the production of organic acids. So it can actually, in some cases, dissolve and create a mining system where it's pushing and diving and then creating organic acids and doing some dissolving at the same time to break through. And they're amazing at penetrating through the soil surface.
Can some of those organic acids also break up bonded elements in a like agricultural soil?
Sure, it's chemistry. And so it's just like you think about organic acid. It might be confusing to some people, and it can be, but an acid is just on this pH scale. And you can think of an acid like an acid. It's a below neutral, so it's a below a pH of 7 at some level. And there's strong and there's weak acids. Organic acids have a wide range of pH scales, but they're typically always below neutral. They're not alkaline, they're not neutral, they're acidic, and they have the same properties of an acid that you would add. This can anyone that's mixed nutrients can relate to this. It's adding a pH down or an acid to your nutrient solution to get things more into solution. And so that's exactly the same function. And they can actually control pH in the soil. They have some effect on pH in the soil that way. It's not a strict chemistry, it's an organic acid. So it has carbons associated with that, which makes it interesting because it makes it a really nice buffering capacity. So, depending on the organic acid, a fungi in particular, may be exuding into the soil, it creates a buffering layer. And buffering means it's pretty consistent. And so, if you know buffering in chemistry, you can have a buffer of 6 pH, or a buffer of 5.5 pH, or a buffer of 7 pH, or even more alkaline buffers. Once you buffer a solution, you can add a lot of different ingredients that might have different pH ranges, but it has a buffering capacity to maintain that buffering level. And so that's what's really special about that. Fungi can occupy a space, and they can kind of control or maintain consistency of the habitat within that environment, not only by just creating physical structure, but by creating a chemical presence as well.
So with growers, as growers, when we make our soil, we try to have like a relatively and like what do you call it, like well mixed soil, basically, where there's no
Yeah,
a
homogeneous.
giant clot of whatever it is. But obviously in nature, your fungal networks are going to have access to like all kinds of stuff, and over here there's going to be this thing, maybe like an animal just died over here, and over here there's some rock stuff like you were just talking about. How does the network kind of decide how nutrients are allocated? Is it from a root kind of sending out a signal? And then that made me kind of wonder about, like, is there like, what's the transportation time from the like rock over here to the root? I was just kind of curious, like, how does stuff move around in this tube network that you're describing?
Yeah, well, it's awesome. And so there's kind of two processes or two things that we'll talk about here based on the question. It's the physical nature of moving something, let's say some nutrition or whatever, from point A to point B. And then there's the decision making of why are we growing that way? Why is it growing that way? And why is it going to mine this area in particular? Or where is it going to grow and why? And I think that kind of comes down to um, a chemical signaling or some cues that the environment can give it. And I'll tell you more specifically for symbionts, for arbuscular mycorrhizal fungi, and maybe there's a network of arbuscular mycorrhizal fungi that are allocating resources to a series of plants. So there's this network, and they're associated with different plants. The arbuscular mycorrhizal work by actually infecting the root zone. So you could think about it almost pathogenic. It's close to being pathogenic because they're infecting or invading, breaking through the cell structure and living inside the root. And then they create these sites, and these sites are actually within the root or exchange sites. And there's in nature, there's always a trade-off. So the plant always gives carbon. The plant's fixing CO2, allocating carbon through the biomass. It's allocating carbon to the root zone to grow. All the mass you see is mostly carbon, but then it's in labile forms and in recalcitrant structural forms. And a lot of the labile or easily usable liquid carbon or sugar can be exuded into the root zone. And it can be exuded to feed these mycorrhizal fungi, and they exchange that through that site, that vesicular site within the root zone. And so there's an exchange. The plant's giving the fungi carbon, sugar, change of carbon, just sugar, and the fungi is extending its hyphal network from that root into the environment, extending beyond the root zone. And in the case of arbuscular mycorrhizal fungi, it's scavenging phosphorus. And so there's a phosphorus for carbon exchange. And so to answer your question, there's always an exchange, but the signal of that exchange can dramatically affect where that fungi looks for nutrition, how hard it looks for the nutrition, and where it reallocates that nutrition to its symbiotic host. See, saprobes are free living. They don't need a living symbiont to grow. These arbuscular mycorrhizal fungi do. And so they're speaking somewhat chemically with the plant to dictate some of those. JR? Sorry.
And so when you say speaking, and we look at the allocations and complicated networks where it's feeding maybe a little tree over here, a little this, and this guy over here, a little that, would you say that is a built-in intelligence? Or would you say it's just random chemistry? How does it know to say, hey, give this guy this, to prioritize? How is it having the knowledge to prioritize feeding others over itself, or even just this guy and not that guy?
Yeah, I wouldn't call chemistry random because interactions are very predictable. And so if you can have predictable input, then you can get some type of predictable output. That's kind of in data science. That's it. Good data in, good data out. Same kind of thing. Well, there's predictable chemistries. You add chemistry A and B, you can stoichiometrically predict output C. And so there's different ways that works in nature. And again, there's a lot of science. It's kind of in some ways new, and this is a new understanding, but there's magnitude and concentrations and chemical makeup. So there's three different kind of ways that plants can signal, hey man, I need more. One of the ways is, let's say from the plant's perspective, say the plant has plenty of water, it has plenty of light, it's not limiting in any way, and there's enough nutrition within its natural root zone area that there's no limiting force, there's no limiting factors. You know, the law of least limiting nutrients, or law of least limiting factor. As soon as there's a limitation, it stops or staggers growth until that limitation is served. Phosphorus might be an example. If there's enough nitrogen and there's enough of these other nutrients, but phosphorus isn't available, then the plants are going to stagger and really focus on phosphorus, because phosphorus has to be in some stoichiometric balance for the plant to grow right. That's for any of these micronutrients or macronutrients, etc. So a plant may slow down the root exudate. It's feeding it because it's trying to get something back. And so it's paying a deposit for a return. It's making an investment. These are making investments, and nature wants a return on that investment, because those investments are expensive. And the ultimate prize is success, which is the ability to live or die or pass off its genome, technically, in biology. So that's that trade-off. Well, a plant will slow down the magnitude of that exudate, and that's one way through feeding the fungi less, where a plant over a little further away might be struggling, and so it will start feeding that fungi more. And so that's one way where there's a direct physical response, which I think it's intelligence. You know, if you get hungry, you eat. If you're getting fed more, you're going to go over to that side. You're going to go over where food's better. That's just nature. And so I think nature is pretty intelligent. There's also chemical structures and different chemistries that might train the fungi to think about what it needs to eat. There's priming that actually triggers bacteria in some cases also. So the chemical makeup and the magnitude are huge cues or signals that allow the fungi to know exactly what organism it should feed or have preference to. There's a lot of studies around that, actually.
And so what about like some of those studies that demonstrated like efficiency, like planning like subway systems based on fungal networks and stuff like that?
Well, I mean, I think that there's a lot of interesting data. What I saw and what I'm familiar with is understanding where there is preferences across different species. That's it. I'm speaking to some of these studies where they plant different species, and some species are really well known to have more root exudate, and some species are known to have less exudate, but they have the same type of fungal symbiont. And so you can actually radiocarbon CO2, isotope label CO2, and follow that through the plant and into the fungi. And then you can actually radioisotope label phosphorus. And so you can have pools of labeled phosphorus that's going back up to the plant, and you can have pools of labeled carbon that's going through the plant into the fungi, and you can see who's exchanging more. And in some of these studies, the plants that are known to exude more actually are getting more phosphorus because they're getting preferential treatment from the fungi because they're feeding them more. And so there's these types of scenarios, and plants have been known to shut down too when they've had enough. They've kind of stabilized and they feel like they're at a good place. Even through the growing cycle, they can reduce their exudate. They want to focus more energy. The more they give out, the more they're trying to give back. But if they're stoichiometrically well balanced, maybe they want that biomass for themselves. Maybe it can have it. Or if there's an area where it's actually gotten stabilized, and so now they can shut that down a little bit. They don't have to give away as much, and they can use what they have for themselves. And that would trigger the fungi to allocate towards a different host in that particular case.
So it's not like they're benevolent individuals who are caring for the love of everyone. They're going where the food is.
Hey, listen, if I were blind, they don't have eyes, man. They don't have ears. What they do have is a sense of how they're absorbing nutrients. And if I, was walking around with muffled ears and a blindfold, and I walk to the right and I'm starving and I don't get any food, no one's spoon feed me anything. But then I walk to the left and all of a sudden someone's starting to spoon feed me applesauce or whatever. I'm staying to the left, bro. And I'm a person, you know what I mean? I'm just naturally gravitating. So I think about it as a pretty simple process. You go where the getting's good. And that's how nature is, like any other life form, you're trying to figure out where you can spend the less energy to get the most bang for your buck. And the chemistry it's just a part of that process.
So I was looking at JR's question. He had a question here about uh, if there's differences in nutrient transfer efficiency between woody soil aggregates. And when I was thinking about his question, I was kind of thinking about how a lot of us these days, a lot of our listeners and people in the community these days, we're building our own soil, or maybe we're like building parts of it or something like that. And so, and we had OK Calix on the show just the other day, or like a couple weeks ago, and he was saying that he just grows like straight compost, which was kind of interesting. And so I was kind of curious, did you see in all your work, is there a big difference when you've got peat moss over here and compost over here and worm castings over here? Are you seeing much difference between what's helping get the nutrients around and getting the nutrients into your roots, essentially?
I spent probably the most time thinking about that question. It was a very interesting question for me. And, I wanted to include like mineral soil to give a contrast of that, for to help answer that question. And so let's think about what these characteristics are. There's mineral soil, like farmers, uh, you know, large scale agro conglomerates. They're farming soil, really, and hopefully you're giving back enough so you don't deplete your soil. We know that happens over time where soil gets depleted because there's not enough of a of a
Crop
of a
rotation.
replenishing crop rotation. That's what comes in crop rotations and stuff. And the crop rotation is mostly to replenish the soil, maintain the structure, and replenish. So we know that. Then you think about maybe a cocoa and a peat, and let's just go all the way to wood chips.
Yeah.
Well, what happens over time in a cocoa or a peat? And we're talking about, let's think about not replenishing. Maybe in the short term, there's enough nutrients in these wood substrates to really give a boost in an actually natural wood environment. You might get a really nice and natural boost because you have a lot of microorganisms. They're going to be decomposing the easily decomposable wood. It's going to release the nutrients that are associated and bound within that substrate. It's going to leave behind recalcitrant wood or substrate, which is less decomposable and has less properties to actually hold nutrients. Well, I've been growing in peat for 3 years just to do a study in gardening. I have these metal rings, kind of like fire rings, and have the same substrate all the way, like 8 of them. And I'll tell you what, the first year, awesome. The second year, and they're left outside, and so they're left to the elements. Plants don't want to grow in that stuff unless there's a nutrition, which I thought was really fascinating. There's more compaction, so there's a physical property there. I think the roots actually took up a lot of whatever was there in the initial bag of peat. And so there's a short-term period where you're going to see a nice boost of taking in all those nutrients. I think that's how to think about it. But without replenishing, those woody substrates need some additives, some soil. And you try growing something in cocoa, especially professionally, without fertilizing it. See what happens. You're going to see the difference pretty quick. You're going to see one that's fertilized and one that's not. You're going to see a twig and you're going to see a full-grown plant. Obviously, soil has a really interesting property of holding a lot of nutrients, and it can release it over a slower period of time. let's say just regular home gardeners that are growing tomatoes and they just have a soil garden and they don't fertilize. Well, you typically see pretty robust results or consistent results year after year after year in that soil. You probably want to put some additives, but the soil will actually hold a lot more. Some of these, it depends on the texture. If there's sand, silt, clay, clay can hold on to some nutrients forever. And soil has a tendency to leach some critical nutrients. Nitrogen's a really hard one to keep in soil. So you're managing some nitrogen in soil, but you're also going to have to manage nitrogen in the woody substrates because you'll use it all really quick and it won't be replenished. And so it was a weird question for me. I think in both cases, it's just not like a one and done. What I think is you probably have more bang for your buck and some fresh wood. There's some stuff, but over time, that's going to deplete very quickly in these wood substrates.
Will these wood substrates provide food in a way that maybe they might not be as attracted to the exudates?
When I think about these wood and the carbon substrates, you think about adding the right kind of carbon to improve soil fertility, and that's really important. And these are these organic acids and all these things. So, uh, higher levels of soil organic matter will actually improve nutrient holding capacity in soils. And so, what I was looking into this a little bit, so what I was kind of seeing was a little counterintuitive to how I would have knee jerked and answered this question. But soil organic matter in wood will have a much higher, typically, water holding capacity. Depending if it's just wood, obviously there's a lot of porosity, and so it could dry pretty quickly. But organic matter will have a lot of water holding capacity. But as it degrades, it's going to degrade the ion exchange sites that will hold nutrients to a higher degree over like a clay soil, which has a ton of really robust ion exchange sites. And it can bind to the metals, and the metals can hold on to nutrition for a long time.
And so the saprophytic are lone stars that are breaking down, uh, and then are the endo uh, having relationships with that broken down materials and making that available? I mean, is there a symbiosis between them? I guess is what I'm asking.
I think there's a relationship from a resource management perspective. Everything's trying to manage resources. Free living saprobes are super effective at, and you hit it on the head, super effective decomposers. They're the decomposers of forests. They're going to decompose rotted trees into the very soft moist. And you see this in Oregon and across the Pacific Northwest, where these huge trees that have fallen, they'll turn spongy over time. Why does that happen? Well, that happens because the fungi grow through there, the saprophytic fungi, and we're talking about the basidiomycetes and the ascomycetes. They're very enzymatically capable, especially for carbon degrading enzymes, and they'll break down all those bonds and just decimate or flatten a forest with any of the fallen trees over time. The endomycorrhizal fungi, or the arbuscular mycorrhizal fungi, the symbionts, they're not free living. They cannot live without a plant host. And so, without a plant host that they can actually symbiote with or grow with, they're not going to exist. The spores will not germinate, and because they won't find a home. And although those spores can live in the soil for year or years, if they don't have that host relationship, they're never going to germinate and they're never going to actually survive. Arbuscular mycorrhizal fungi, IAM fungi, are not very enzymatically capable. You're not going to find them degrading or decomposing forest systems with logs and really really recalcitrant material, but they could gravitate towards pools of available nutrients when they're extending beyond the root zone of their symbionts to capture phosphorus. And so as that decomposition is happening with the saprophytic fungi, as it releases any phosphorus materials, it will sense that it could grow that direction. You find a lot of spores arbuscular mycorrhizal spores. You'll find those in those materials. But it's typically because there was a relationship with the plant that was close by, and that fungi is growing in there trying to scavenge. In this particular case, in the case of arbuscular mycorrhizal fungi, phosphorus. The other case, there's those two types of symbiotic fungi, the ectomycorrhizal fungi, and we can think of those as not endomycorrhizal fungi, but ectomycorrhizal fungi, meaning they grow along the surface of the roots. They actually cover the root zone with hairs. And those ectomycorrhizal fungi are associated with wooded plants. And so with shrubs and trees. And so you're going to find those groups of symbiotic fungi growing along the root zone of those woody plants. Or arbuscular mycorrhizal fungi will grow with the grasses and some other plants and a lot of the annuals. And so that's the difference between arbuscular mycorrhizal fungi and ectomycorrhizal fungi. Some ectomycorrhizal fungi are also free living fungi, but there's a whole group that are just associated fungi that are just saprobes, free living fungi. They live to go decompose a tree log. And so that's what they do. And they'll break that down using a lot of organic acids. They'll use a lot of penetration, a lot of carbon degrading enzymes, and they'll turn that tree log into sugar that they can absorb. And that's what they live for.
So we've had several. I've got a baby that's just roaming around behind me. So if you guys can,
that's okay.
I'm sorry. He's just having a good time.
That's so good.
But anyways, so we've had several episodes for the last handful of months where we've talked about IMO collections. So like you got some rice, you put it out somewhere, and you're collecting your local neighborhood mycorrhizae or whatever. So are you collecting like certain types of fungi when you're doing that? You mentioned earlier you were talking about the different types kind of are coming from different types of material in a way you could say. Is that kind of more what I'm collecting kind of based on where I place it, or is it also I'm just like collecting very specific types of species, I guess, or types of mycorrhizae or fungi?
My understanding of it, I've never done it, so I can't give you a direct account of some direct experience. But my understanding of that practice, and I did a little research also, so I kind of found what other people were finding and actually quantifying, is based on the bait trapping and everything you're doing for the indigenous mycorrhizal or microbial organisms, and we're mostly collecting fungi. I think to a large degree those are biased towards a free living saprophytic fungi because there's just going to be a lot greater concentration of those. You're going to find spores, you're going to find hyphal strands growing through all of these materials, and they'll be attracted towards that. The bait in many cases for arbuscular mycorrhizal fungi would have to be a living plant root that has an association with a specific group. And so you kind of have the wrong bait to be a super robust and capture a bunch of indigenous arbuscular mycorrhizal fungi. Not that you can't capture spores, because the spores will be in that material and they'll sporulate as they're growing, but they're only sporulating once they're associated in active growth with an active host plant. And so the number of spores is probably way less than the number of spores and species you're going to find from the free living saprobes, which is a huge group of different fungi.
So when I'm collecting leaf mold and it's loaded with a diverse group of mycorrhizal fungi, most of those are going to be in the saprophytic form.
Yeah, and I just think about those as free living soil fungi. And then mycorrhizae are the symbiotes. And so I wouldn't say exclusively you're going to find any one thing. I've collected a lot of soil samples, and I always got a signal of some forms of mycorrhizae, but it was a minority of what I captured just because of the environment. And if you're sampling very meticulously, destructive harvesting, well infected or well colonized ectomycorrhizal fungal symbiont or arbuscular mycorrhizal, you're going to get a lot more of those, but you're going to have to go where their hosts are to really collect that and get a lot higher concentration of the symbiotic fungi. That's my understanding. And when I looked into that, because I thought that was a great question, you're going to find spores, but then once you find the spores, until you can associate them with a plant host, the spores will never germinate. They're just not going to germinate by themselves, for the most part.
So we just did an episode just the other week with from Growcast, Jordan from Growcast. Sorry, I can't talk today either. So anyways, Jordan, I'm sure you know Jordan. So we were just talking to him.
He's great. He's super good, isn't he?
Yeah, and
he's
so
really
we're just
good.
talking about foliar applications. And so can you talk about, so people love to just throw stuff at their plants, and the more the better. Is that kind of here, right, where I'm spraying this fungi onto my plants? But it's like different here, right? It's not tapping in. There's no like Eiffel network in the sky that it's tapping into. The leaves are tapping into, or whatever. So how does that work? How is it helping my plants?
I don't know. I really don't know. I mean, because I'll tell you, I'm a soil guy. And so I think about how microbes, soil microbial organisms, bacteria and fungi interact within the root zone. In some cases, I can understand how they might colonize a leaf. Bacteria more so, pathogens for fungi, because you think about things go towards food. Period. If you can find a food source, then you can find something that wants to eat that food source on that food source, and that's how that works. So most of my studies have been around these root symbionts, or these root associated bacteria and fungi, whether they be ectomycorrhizal fungi, saprobes that live around roots too. They don't avoid the root zone, but they'll extend further, and they'll live off of dead plant material also. I mean, they'll grow right through a log. I don't know. Do you have polypores and stuff where you guys live, right? The big shelf fungi.
Mm-hmm.
Those are crazy, aren't they? Those are the evidence of these fungi just eating up a tree. And there's these big, I don't know if the audience has seen them, but they're hard. They're very woody. And they're getting all that carbon from dead plant material and allocating it into their biomass. And so there's that, and the arbuscular mycorrhizal fungi, which obviously won't grow, to my understanding, anywhere except in a root zone.
So one of the things that they tell us when we're trying to apply this in a indoor setting with these fungal products and applications, we want to apply these very early stages. Can you kind of talk about why that is, and also kind of talk about the moisture levels that we're trying to maintain and ways that we can know our mycorrhizal networks are doing their job?
Sure. I want to say one thing really quick. I want to learn more about this foliar stuff. I'm not saying yes or no. I don't want to poo-poo what anyone says, but I will say I just wanted to be clear on that. I don't understand how fungi really live above ground to any capacity. But what I will say is it's very clear that fungi, I'll answer the second part of your question first, JR, are aerobic, meaning they do not like anaerobic conditions. They cannot grow in anaerobic conditions. And if you create an anaerobic condition, then you will not have successful fungal colonization or growth. And so you think about the water holding capacity, or the water holding capacity of the volumetric water content of the soil. And typically you want to maintain, 20 to 30, maybe 40%, which is if you're filling up the volumetric water capacity of the pores in the substrate, whatever that substrate is, it can be from 50 to 70%. And so you have a physical matter, you have air matter, then you have water spaces within a substrate, and you want to have that nice balance. And you want to get to that point where water's not limiting, but there's plenty of air And so if you think about that range, there's still adequate aerobic conditions for the fungi to occupy that space without going anaerobic whatsoever. And the timing of applications, that's a really good question too, and it depends on the microbe. But if we'll talk about arbuscular mycorrhizal fungi, typically, and this is just how I think about this early on for the colonization, it typically can take up to 2 weeks for a mycorrhizal spore to sense an environment, meaning a hospitable environment, meaning where there's a host plant that it wants to grow into or wants to be symbiotic with. Then it will germinate. That's very conceptually just like a plant seed, germinating and creating an initial sprout where it's going to root down and then create an above ground shoot. In this particular case, a spore is just going to shoot out a hyphal strand, and that hyphal strand is going to look for a host plant in the case of arbuscular mycorrhizae, and it's going to look to try to impact that host root and then penetrate through the cells, in between the cells, grow in between, and start creating arbuscular sites. And so that can be a 2-week process, and it's a probability game. The root should be close, and there should be a lot of spores in that area to maximize the colonization. I would say early on because of that timing, JR.
And so if you're overwatering and you're kind of suffocating things, are they going into a natural dormancy state? Are they drowning? Or when you got aggressive drybacks, are they also going into a dormant state?
I would say if anything, you create more pressure for fungi in a saturated environment because you create more tension of osmotic stress. Fungi, hyphae, are built to withstand, and some more than other saprophytic fungi, and some saprophytic fungi that live in desert systems are better than the ones in some cases that grow in mesic systems. But these hyphal strands are tubes. It's like a straw. I don't have a straw here, but it's like a straw that can withstand a lot, and it can sit there and it can transport nutrition, and it can transport water over dry patches, from wet patches over to dry patches within a broader sense. At one point, the fungi, a single genetic, unique genetic species of fungi was the largest organism on Earth. You guys heard that, right?
Yeah, here in Oregon, right?
Was it in Oregon or Michigan?
I thought there was one in Oregon that was acres that I thought was one of the largest organisms on Earth.
Pando or something? Pendo? I can't
remember.
Something.
Yeah, I think it's in Eastern Oregon.
Yeah.
Fair enough. There was another case in Michigan a long time ago. The largest single organism now is actually a tree, I think. It's aspen grove. And you know how they're a unique single organism. They create rhizomes and they just grow everywhere.
Yeah.
So honestly, these things do a really good job of withstanding, for the most part, dry conditions. There's extremes where some fungi can just live forever. If it's too dry, they'll sporulate, they'll go into a dormant state and just wait for good conditions to come back up. Then they'll revegetate, meaning create new hyphae. But fungi can usually do a pretty good job if there's a part of the mycelium or a part of that greater body that has a resource somewhere that can reallocate, and it allows them to stay pretty robust for a long period of time. Also, these hyphal strands, they're not very robust to physical destruction, meaning tilling is really bad in farming because it just immediately obviously rips and breaks apart these hyphal strands with these mycelium, breaking that network. But undamaged physically, they can maintain their structure. And if they maintain their structure, they maintain their function. And so drought and rewetting, they're actually better at that than bacteria, where bacteria can be very, depending overall, I'm speaking generally for bacteria, can be very susceptible to strong drybacks and strong rewetting events. Fungi have to respire. And so they will go into a suffocation mode and they can't perform their electron transport chain at some point. And the excessive water, will knock them out a lot sooner than excessive dry conditions. I would say also, lastly, mycorrhizal fungi, especially arbuscular mycorrhizal fungi, are more sensitive to environmental conditions and stress than like saprobes in general. But you think about, or you hear this a lot with arbuscular mycorrhizal fungi, that plants that have arbuscular mycorrhizal associations are a lot more drought and stress tolerant. I don't know if you've ever heard that, but you can read that almost everywhere. That suggests that these fungi are able not only to withstand those environmental extremes, but they can also transport some of that resilience to the plant, to their host plants.
And so now a lot of times in, commercial settings or even in home settings, people are just hammering the CO2 to get efficiency to match their ppfd. Is that hammering of a high CO2 environment, is that impacting your fungal networks?
It impacts them indirectly. And it kind of goes back to what we talked to. There's this food source, and there's this need for the plants to have a certain amount of resources, stoichiometry, the balance of all the macro and micronutrients for them to grow. Um, in some cases, additional CO2 will allow the plants to allocate more carbon through the root zone, through exudate, and feed those fungi more. If they're feeding the fungi more, they're going to stimulate fungal returns and they can get more out of it. Likewise, if the plant has everything it needs and it's allocating the CO2 and it actually extends its root zone, there's more habitat too, obviously. If you have more carbon, the plant's are creating more below ground root zone habitat. And so it can create a nice environment which will stimulate or support more fungal below ground root symbiotic fungal habitat for it to grow. But if the plant has everything and it's getting fed also, it could just take in that CO2 and hold on to it and just grow upward if it has all the other nutrition that it needs. And so it can actually go either way, but it's a function of whether it's passing on those resources to the fungi or if it's holding on to them to grow.
Awesome. So sorry, I've got dogs now going crazy behind me. So JR, you're gonna have to go in with that last. I've got a question to throw in after this one.
And so a lot of times we get products from shops and from places, and a lot of them come in a dormant state, and there's not a lot of production going on. Is it the process that makes it such a hard thing to produce, or is it just not enough money for people to really jump in and want to hammer it out?
Are you talking about fungi?
Yeah, like say I buy
Yeah.
my Mycos from Extreme Gardens. I'm dusting my root zone. There's not a lot of facilities making mycorrhizal products, right?
Yeah, totally. Yeah, there's a really good reason for that because it's hard for them to make. And it goes back to what we were talking about before. Typically, I know one place that claims it doesn't need a plant host to grow mycorrhizae, but typically, to grow certain strains of mycorrhizae, or muscular mycorrhizae, you need that plant host. And if you need that plant host, all of a sudden you need a place to grow those plants. And then you're inoculating the roots, and then you're allowing for that colonization to occur in the root zone. And then there's typically a shaking and a screening process to harvest the mycorrhizae. And harvesting the mycorrhizae, typically they're harvesting hyphal strands and spores. It's a really interesting detail. If you look at these products, it can be caught as propagule units, a number of propagule units, and propagule units can either be a hyphal strand or it can be a spore. And those might have very different success rates, but it's all clumped into one. And so it's a good question to ask: are you selling me spores, or are you selling me propagule units, or are you selling me both? Because that has implications for what you're actually buying. Typically, it's the propagule units are including these hyphal strands and including the spores, and however long the product's been around indicates how successful, especially those strands, those hyphal strands, will revegetate. So these hyphal strands have septae, and so they're actually segmented into little groups, and they can close off and maintain their function for a while because they'll maintain the structure. But if they get too dry, the ends will kind of dry out, and then all of a sudden it's just a piece of biomass or dry straw. So it doesn't have the ability to rebound like it would if it was newer, if that makes sense.
Yeah.
So
Interesting.
the infrastructure and the labor and everything it takes to harvest these fungi, and I think that what you find is there's probably just enough places that can supply that global demand that no one else really wants to get into that game. And so you find, honestly, probably 7 producers worldwide that produce it for everybody.
That's so interesting. Well, I'm gonna throw in a question that was on my mind, and if this is outside of your wheelhouse, I just let me know. But I was gonna ask about Botrytis, which is another type of mold. But it came to mind in this context because right now we're recording in the beginning of September. I was just watching a outdoor grower. He was talking about how we're going to get rain on the West Coast, and he was talking about how the way that he tries to limit the risk of botrytis in his outdoor grow is he doesn't water his plants as much, and he kind of tries to keep the top of the soil drier. Is that a thing that you want to keep, like, kind of the top of the soil drier, so that a botrytis mold doesn't kind of grow there or live there? Or is that kind of, I'm just curious if you have any thoughts on this topic.
I don't know. I think about botrytis being air dispersed.
Right.
And so it's in the air, and it's not in the air as an active fungi. It's in the air as a spore, or the seed, and it's going to land on a food source. And it's no different than below ground. It's going to land on a food source. It's going to sense a food source, and then it's going to start growing from that seed, or it's called the spore form, and vegetate. And it's going to create hyphae, and it's going to extend out. And that hyphae is what actually penetrates and hooks in to that substrate. And the substrate in this case is your active growing plant, and it's going to start degrading and absorbing, exuding enzymes, maybe exuding some organic acids, turning that leaf into a structure where it can absorb. So these things don't have mouths, and so they have to create a liquid labile food source that they can absorb through their cell walls. And so they just create a liquid source that they can exude in. So if you maybe, but honestly, I think that if that process reduces the actual airborne spores, if maybe there's a ton of spores on the ground there, and yeah, I don't know, but that's how I think about it. If it reduces the transport mechanism, it should reduce it. I can't speak to that.
Yeah, it makes sense.
I was listening to someone talk about the life cycle of powdery mildew, and how in the wet season it's in the ground and it's spreading and breeding and growing. And then as the dry season comes, it's kind of sporulates out into the air. Then it lands onto a host, and then it keeps that cycle of life constantly continuing. And so I'm almost wondering if, if you know the life cycle of how it's being bred and sporulated out, maybe you could help prevent. And I do know, for like in the case of powdery mildew, one of its natural enemies in the soil is bacillus bacteria, and also nematodes will actually consume the powdery mildew in the soil before it actually gets a chance to kind of sporulate and do its thing.
Totally. I mean, that's exactly right. It's no different than insects. The way we kill insects and do IPM is you understand the life cycle and you cut it off at the life cycle. It's no different. One thing that'd be fun. I haven't thought about this in a long time, but back in fungal class and fungal biology and everything, way back, a long time ago. If have you ever studied, this isn't relevant for cannabis, but for growing and for life cycles, this is one of the cooler life cycles is the life cycle of corn rust. Have you ever had anyone talk about that? It's a crazy, so it reminded me of what you were talking about. It's a crazy 2-year life cycle, and it goes through half of its phase the first year, and it's undetectable. Then it goes to the second half of the phase, and it'll just decimate. And so you have to figure out how to find it in that first half of the phase when it starts to come up. Otherwise, you're gone for year two. And so, it's fun just on our own time or on your own time if you're interested. You can look at the life cycle, and you can actually see, just to answer that question really well, how farmers have learned to cut off the life cycle. And that's what you do. You find out where to cut them off. For a lot of sucking and sap sucking insects and whatnot, they have these early stages, these larval stages, they have juvenile stages, and they have young adult stages, and they have these breeding reproductive stages. And once there's a bunch of eggs and they're growing, they can get to an adult reproductive cycle pretty quickly. And so you have to cut off these juvenile stages before they reproduce. And so you have to do that not only once, because there's going to be, if you have an infection across a wide different ranges for these insects of different life cycles. And that's why you have to hit it multiple times, because you're chopping off the life cycle until you kind of have all the eggs that have hatched, and so you can get that, so there's no more eggs that are being produced to continue the population life cycle.
Awesome. Well, we really appreciate your time today, Colin. Before we let you go, where can people learn more about you? I know you have a Instagram. Are there websites? Are there other things? Where can folks keep in touch with you?
Yeah, you can always go to Instagram, Colin W. Bell, the real Dr. Colin. You can always go to my LinkedIn. I'm working with a lot of great companies. Mim Horticulture, I'm still working with them. They're a great company, and they've been super supportive, making a lot of cool stuff. And there's always a lot of interesting things happening in this industry. I don't know, needless to say. And so we're learning and growing, figuring out how to navigate interesting times. And I love that we're all still here, working together to try to figure it out.
Yeah,
Wow.
I agree.
Hey, Dad. Golly, you
Yes.
got your hands full, bud.
Yeah, for audio only listeners. Yeah, I've got a baby now, popping up on the screen. Yeah, he was doing pretty good for the most of the podcast, but now
Yeah, he
he's
did
popping up
great.
to say hey.
Yeah, totally.
Well, thank you once again. We really do appreciate your time, Colin. Thanks, JR, for putting together today's episode. Jasper's sad to see it end. But we really appreciate everyone's time. We appreciate everyone who's subscribing over on Spotify. Appreciate your support. Leave us a comment on Spotify. We'd love to hear from you over there. And then otherwise, we hope you have a great rest of your week. And JR, as always, growers love.
Growers love.
Peace, everyone. Have a great rest of your day.